So, hello everybody. My name is Sven Borgos. I'm working in SINTEF in Norway, which is feedback. A non-profit CRO and one of the partners in the UNCL, and we contribute to the physical-chemical and to the in vitro tox characterization. As you've already heard, we have an established characterization cascade, which is very heavily built on what they've developed in the US, in the NCL. However, we want to do better as well. We want to improve the whole cascade, and that can be improvement on parameters like robustness. It can be things like speed. It can be things like cost. It can be things like sensitivity to pick up, for instance, well, endotoxin contamination is one obvious thing to look for. And it could be entirely novel endpoints, giving us information that basically wasn't available before. Now, this means that we have to focus on applicability. It has to be useful. That also means that we are not going to pick up on the very cutting-edge science, the very most novel prototypes. That's not us. And it also means that in order to be useful, we have to consult the stakeholders about their needs. Now, the kickoff was in 2015 for EUNCL. In theory, we could have started here in terms of capabilities. Now, because we have this wonderful friendship with our cousins in the US, we could get the flying start and start quite high on the curve. And then, in the worst case, we would asymptotically approach their level here, and that would basically be it. But we all strive to do better. So the US they are launching their version 2.0 of the NCL, and we are trying to do the same thing by doing these assay improvements within our project. So hopefully this curve here will keep on growing into eternity. Now, the process for improvement is very basically like this. Technological opportunities means novel technologies mostly. So we identify some candidate technologies. They can be completely novel, or it can be improved variants. That's kind of the brainstorming level of this. Then we have to integrate the needs of the stakeholders, and typically that's regulatory needs. So we can prioritize the rather large number of possible candidates here into a prioritized set of technologies. Now, from those, we perform initial testing in the lab, seeing if these are feasible to apply to nanomedicines. And if they are, we can take them further into interlab testing and early, validation is a big word, but qualification anyway, so that we can demonstrate to regulators and other stakeholders that they are suited for the characterization. And then we can take them into the standard assay cascade. I'm going to talk about 3 technologies that we identified in the first iteration of this process that I showed you. They cover basically all parts of the cascade. Nuclear magnetic resonance, NMR, is currently not a standard part of the cascade. I'm sure many of you are familiar with it. It's a rather mature technique for small molecules and for proteins. It's been around for really quite a few years. It's a very general detection principle. It's not specific to one type or family of compounds, because you're looking at the nuclear spins of basically any kind of nucleus with a net spin, protons most typically, 13 carbon, 15 nitrogen. It is untargeted. So if you have a molecule, it will give you, in principle, information on all parts of that molecule, all the atoms in that molecule at the same time. So that's kind of in opposition, for instance, to optical techniques that need a chromophore or a special functional group to give you a signal. It is sensitive to the chemical environment. So a molecule encapsulated and a molecule free will, in theory, give you different signals, which is very useful when you are actually trying to encapsulate things. It is relatively low in sensitivity compared to a few other techniques, and that might be the limitation. That means NMR has some specific applications. Potential applications in nanomedicine, API quantification, ligand quantification, and as I mentioned, localization, which is very, very interesting. Here's a case that we did on NMR. We were going to quantify the API in a liposomal formulation of doxorubicin. Now here's the structure of doxorubicin. We'll label the carbons for you so that you can correlate them to the NMR signal that I'm going to show you. And we identified most of the relevant peaks with the expected doxorubicin concentration. It's a nice red solution. And then there was something else. There was something in the NMR spectrum that wasn't really supposed to be there. So you can see most of the signals here can easily be identified in the doxorubicin molecule. But this one here, this signal here, is not really supposed to be in that molecule. And it wasn't in the standard. And it turns out that we were actually not given doxorubicin, we were given daunorubicin, which is a very close relative, with a slight change on this carbon here. And that explains the NMR signal, and it fits perfectly with the daunorubicin standard. The amount was the right, but the actual compound was not the right one. So that's also something that could happen. The second technology I'm going to talk about is more on in vitro cytotox. MTT, I'm sure a lot of you are very familiar with that. It's more or less a standard for cytotoxicity. This nice purple one here. However, it has some interference issues, and that was identified for nanomaterials already quite a few years ago. This is from 2011. GO is graphene oxide, GS is graphene, and this is the MTT assay. This is a variant called VST8, which is based on a water-soluble analog of MTT. That is technically much easier to work with, and it's much less prone to interferences. So it's not a dramatic change, it's more a variation on the MTT assay. It goes yellow rather than purple, that's very visual. And just to show you some very initial results on that, this actually demonstrates, this is not a nanoparticle, this is digitonin, which is a commonly used positive control in cytotox assays, but it shows you pretty well the interference issues. Now, here's the red one, here's the MTT curve, and you can see that the dose response curves are they're not very nice for MTT, but for the VST8 and, well, another formulation just called CCK8, which is basically the same chemistry, you can see you have these really nice, sharp cutoffs. Then we tried it on nanoparticles that we have in-house in SINTEF, which is cyanoacrylates. Now, in a way, these are not the most perfect ones to demonstrate interference because they don't really give a strong interference with MTT, but the curves do show that the VST8 assay works very well for these types of nanoparticles. And these are on liver cell lines and kidney cell lines. This is what Adriel already showed, but I'm going to skip straight to the MTT and VST8 part of it. MTT has massive interferences. VST8 gives results that are much more in accordance with the high content data. And there's much better coherence between the two replicates as well. So basically, we are quite far in the process of establishing VST8 as an alternative to MTT by doing interlaboratory studies on this one. So that will be the first one to actually go into, that will be for us the first novel technology to go into the standard cascade. Now the third technology we have been deciding to look at is flow cytometry. Now, Flow cytometry can provide you with really a lot of additional information as compared to just cell viability. This particular example here is on the lopinavir, which is an HIV protease inhibitor. There are several studies showing complications with lopinavir. So we did a comparison of free lopinavir and solid drug nanoparticle encapsulated lopinavir. And Neil, who did these studies in Liverpool, he looked at reactive oxygen species, caspase activation, mitochondrial membrane polarization, and apoptosis. And this was all done by flow cytometry. I'm not going to show you all the data, but just to know that these could all be done with the flow cytometry technique. And he looked at human T cells and monocytes. This is the membrane polarization data, and you can see here that you have the cells only, the negative control. You have the free drug, and you have the encapsulated drug, and you have the positive control, which is camptothecin. And you can see really clear differences, especially in this case, and you can see that the cell lines respond in a different fashion to this encapsulation. These are the THP cell line, and this is the CM cell line. Apoptosis, less dramatic differences, but very easy to gain information on apoptosis from flow cytometry experiments. So really in a relatively easy setup, well, easy, everything's easy if you know it, but flow cytometry is a pretty mature technology. You can really gain a lot more than just viability data from your toxicity experiments. And so to try to conclude here, what we're trying to tell you is that we are committed to not only perform existing assays as a routine, but also to try to push the envelope a bit, try to contribute more to you as stakeholders in terms of characterization, so that we can, in a joint effort, we can bring more nanomedicines into the clinic and to the patients. Thank you.